Hi, everybody. We are ready to get started now. Welcome to an introduction to environmental DNA monitoring for amphibians and reptiles, otherwise known as eDNA. I'm Jen Williams, the National Federal Coordinator for Partners in the Amphibian and Reptile Conservation. I'm going to go over a couple of logistical items really quickly before we start the webinar. If you're interested in asking any questions, please type them into the questions box and not the chat box. We like for folks to use the questions box because we can then export all of the questions after the webinar. And so if there are any that we didn't get to during the webinar, we're happy to answer them afterwards, either through email or we'll notify you of another upcoming webinar that will answer your questions. We are recording today, and this webinar will be posted to the Partners in Amphibian and Reptile Conservation website, and that is at parcplace.org, PARCplace.org, and I'll put that website in the chat box. So a little bit about the speaker. Steve Spear is the Director of Wildlife Ecology at the Wilds in Cumberland, Ohio. Prior to his current position, Steve worked as a conservation scientist for the Orion Society for almost 9 years. He received his PhD in 2009 from Washington State University. He has an MS degree from Idaho State University, and his BS is from University of Richmond. His work focuses on the ecology and conservation of wildlife species, with a special emphasis on combining genetic techniques with field ecology. So thanks to everybody for listening in, and take it away, Steve.
All right, thanks, Jen, and thanks to everyone for joining in. And I'm excited to talk for the next 40 minutes or so about eDNA. And my goal with this webinar is really, the main audience is geared toward folks that don't know much about eDNA, or have heard a little about it, or maybe read some papers and are interested in getting involved but don't know the entire process, or have lots of questions about how it works when you're trying to use it for survey and monitoring and those sorts of questions. I'm not planning to go into super in-depth into any one topic, just because it would be, I could talk literally all day about that. So I'm trying to cover a lot at minimum depth, but I do want to leave time at the end for questions, and that way, folks can tailor what their questions are. And obviously, as the title indicates, I'm focusing on reptiles and amphibians, but the methods could be applied to any tax. So just the examples that I'm given are primarily from reptiles and amphibians. Let's see. Okay, so the first question is, what is environmental DNA, or eDNA, we use for short? And with what I'm going to talk about, we're thinking about it in a more specific way, but just environmental DNA in general is really any situation where you're being able to collect DNA, extract DNA from a source that doesn't come from having to have your hands directly on that animal or plant or whatever you're looking at. So eDNA, from that broad definition, has been in place or people have been doing those sorts of studies for a number of years now, and that includes things like scat, hair. Scat and hair are probably two of the most common things that were pioneered really in the 90s and into the 2000s, and then other things like eggshells, shed skin from snakes, feathers, etc. So there's a whole host of sources of eDNA, broadly speaking, that don't involve putting your hands on the animal. However, all of these examples I'm showing here still require you to find some sign of that animal. So even though you're not having to catch that bear or cougar or whatever and sample it, you still have to, put out a snare for it to go by or to look for its scat. And so it's still a bit of a scavenger hunt. What most folks these days, when they talk about eDNA, they're referring more specifically to what I call aquatic eDNA. So getting DNA from the environment of water.
Hey, Steve.
Yeah.
I'm sorry.
Can you hear me okay?
Everybody's saying that they don't have any audio. I'm not sure. Oh. Yeah, we started the broadcast, and I don't know what is going on.
I mean, obviously you can hear me okay, so it's not my phone, huh?
Okay, you know what? Some people can hear and some people don't. Okay, I'm sorry to interrupt the webinar. Let's just go. Sorry about that.
Okay. Yeah, hopefully other folks can figure it out, and hopefully my volume's okay and everything.
Yeah, it sounds great. I'll just say real quick to everybody, well, I guess the people who can't hear. Let me just put something in the chat box. Sorry about that, Steve. Go ahead.
Oh, that's okay. So aquatic eDNA is really getting eDNA from water, and that was a bit of a game changer because you have to go to the water source, but you don't have to hunt down for any specific sign from that animal, for instance. And really, the proof of concept study for what we do now with aquatic eDNA for vertebrates or multicellular organisms was a study back in 2008 by Fistatola et al. In France, looking at what there is invasive bullfrogs. And what they did was they collected a small amount of water from ponds that they knew had lots of bullfrogs from surveys, sites where they knew bullfrogs were there, but at low density, again, based on previous survey data, and sites where the best survey efforts indicated no bullfrogs. And what this table is showing is they then extracted DNA from that water and amplified it specifically for bullfrogs. And so this is showing the percentage of samples that showed positive for bullfrogs. And what they found was that if you have high bullfrog densities, almost all the time, you get a positive signal of bullfrog DNA. The ones that had low density, and what this meant by low density is like 1 to 2 bullfrogs, you still, not as much, but you still consistently picked up bullfrog DNA. And encouragingly, spots where they were pretty sure there were bullfrogs, they weren't getting false positives. It was 0. So this was really the first paper that generated the field of aquatic eDNA for things like herps and spurred a whole lot of studies and showed that it could work without having to sample a ton of water. And so now, with a cup of water, you can potentially detect all sorts of members of the aquatic community. So, things that are semi-aquatic like water snakes to things like hellbenders, crayfish, trout. I don't have it up here, but even things like deer, if they're drinking from a water source and their saliva is going in the water, you could potentially pick them up. And so it's become a pretty powerful survey method for detecting presence, as I'll talk about further. I'm also going to mention just a little bit, not anywhere near as much depth because the studies on this is really in its infancy, but it's something that a lot of people ask about, and for good reason, particularly with herps, which tend to spend a lot of time underground, is getting eDNA not from water, but from soil or sediment. And this slide is showing that, yes, at least in some cases, it's possible. This was one of the first studies, again, looking at vertebrates, terrestrial vertebrates getting soil eDNA. And this was from a game park in Europe, so elephants, tigers, ostriches. And I'm not going to go into the details of it, but basically the black areas represent where they picked up DNA from those animals. So I'll talk about this more in a herp context later in the talk, but that's another emerging direction for eDNA that doesn't directly involve water. So I'm going to transition into some of the specific methods of how we actually collect and amplify eDNA, and then I'm going to talk about some of the case studies. So the specific methods can vary based on the study, and different labs have come up with different specific ways of collecting, extracting, and amplifying DNA. So this is by no means an exhaustive review of every method, but these are some of the common ways, and certainly what I do in my lab. In general, the two main ways are either taking a small water sample and centrifuging it so that anything in that water sample goes down to the bottom, and then you can extract from that precipitate, which would have all the cells and DNA. So this is actually the initial bullfrog paper I talked about. This is what they did. They used 15 milliliters of water for that. I know Fish and Wildlife Service do that a lot with some of their eDNA sampling. It does require having some of the right equipment, like you need a centrifuge that can handle at least 50 mil tubes, which is not in every lab, in fact, not in my lab. But it's a way that's been effective when it's been tried. The other way that's probably more common is filtering the water. And so you run some amount of water through a filter, and then you actually extract off of that filter. And that's typically what I do with eDNA samples that I collect. There's a number of papers that have looked at what filter types are best, what pore size is best, and to a large degree, it depends on the exact study and what organisms you're trying to isolate from. The range of pore sizes for the filters range from 0.2 to 5 microns. I tend to use 0.45, but there's certainly been good examples of larger pores that have worked. That can be really helpful, particularly if you have turbid water where these filters get clogged up very easily. As I mentioned, the centrifuging water, a common amount is 15 mils. You could go a little higher than that, but you can't do too much water when you're centrifuging it down. Again, it depends on the study and how turbid your water is and what you're trying to detect. But the range for the amount of water that typically gets filtered is 250 milliliters all the way up to 10 liters for some studies. When you have really nice, clear river water, you can get multiple liters. In more realistic cases, it tends to be more 250 milliliters to a liter, excuse me. And if you're dealing with really turbid water, sometimes getting 250 milliliters to go through one of these filters is a real struggle. But that's kind of the general range. This photo here kind of basically shows the basic requirements, at least for the filtration method. You can do it as simply with a hand pump that you can buy from any auto parts store or Amazon. And you hook it up in a flask, and over here are a stack of filters that you put on top. I'll show some more pictures a little bit later. But in a sense, it's pretty simple to collect this water. It's simple, but the most critical thing is not necessarily the actual equipment, but making sure that equipment is clean and you're not transferring DNA from one sample to another. Because eDNA is very much low copy DNA procedure. It means that even at its best, there's not going to be much there. So if you inadvertently spread DNA from some other source into your sample, it's going to probably form a large part of what's in there and easily contaminate your sample. So we have a number of quality assurance, quality control protocols that are essential if you're both collecting and running eDNA that every study should keep in mind and follow. If you're using a lot of equipment to collect your water, like let's say you have to go out in a boat or use nets, buckets, make sure in between every site or depending on your study questions, even every sample, that you thoroughly decontaminate with bleach or something else that degrades DNA, your equipment between sites, if it's a problem that if you spread it. Now, if you're taking multiple samples within the same river and you don't really care because you're trying to have those samples represent the river as a whole, then it's not like you need to clean all your stuff in between each sample, but certainly when you're moving between sites. So I mentioned bleach is a common way to clean gear. DNA away is another one that I use a lot, particularly for forceps, which we use to move the filter into a tube, which I'll show in a moment. DNA away is, if you were using large quantities like we typically do with bleach, would get really expensive, but it's definitely effective. And then there's other disinfectants like Virkon Aquatic that one could use. What's important is something that will degrade DNA. So as far as collecting your water, I'm focusing mostly on filtration here. For the centrifuge method, obviously you're collecting the water in some container, but then you're moving to the centrifuge. But if you're filtering it, you can do it in one or two ways. This picture you can see on the left here that my mouse is over is showing actually filtering at the site. In this case, instead of a hand pump, they're using a motor, which you can certainly do. It makes things a lot faster. But you can see he's collecting the water in the cup right by the stream and then running it through right there. You'll also notice that in both these pictures, they're taking care to not get into the stream. And if it's possible for your study question to sample from the banks, that's certainly preferable because then you're less worried about moving DNA between your equipment, although sometimes certainly you may need to get deeper into there. So that's when you're sampling at the site. If you're sampling, say, in a lab, which I'll show in a second, you can take a clean bottle, obviously either one that hasn't been used before or that's been thoroughly disinfected, and collect your water source. And you can see everybody's wearing gloves. That's also part of the protocol that you want to use. and obviously change gloves between sites. Another really important aspect is having negative controls at multiple stages, but starting in the field. So field blanks of deionized water or some water source that you are 100% confident would not contain DNA from your target species need to be run through using the same filtration system as you do your unknown samples. This is a way to make sure that the way you collect your samples and the way you run through your samples aren't introducing contamination on their own. The general rule of thumb is about 10% of your total unknown samples. It'd be good to have field blanks. Obviously, you can adapt this to your study system, particularly if you've run a lot of samples and you get a pretty good idea of what your error rate may be. For instance, I would recommend, if you're just starting an eDNA study, doing a field blank for every site you sample. So let's say you take 3 replicates at a site, taking a field blank with that, even though that's more than 10%, to make sure that your protocols are staying clean and you're not contaminating. Then as you run a lot of samples, if you show, okay, you know, my negatives are always clean, then you can reduce the percentage, say, to that 10%. So it's up to your judgment, but realize that if you get a field blank that does show a positive, anything that you've run before that field blank, you have to basically consider contamination. So you could lose a lot of samples if you do it too infrequently and you have contamination. So I mentioned you can collect the water and bring it into the lab. So here's an example. It's basically the same setup, just a little bit more comfortable, I guess. This is the motor is not in view of this picture, but this is using a motor to run it through. And again, you pour the water in the cup. At the bottom of that cup is a filter, and it runs through. And you can see that's at the bottom. That's the filter, and it's collected some of the sediment that's in the water. In this case, this isn't too bad. It's not too clogged up, but they can get really clogged, and it can be difficult to filter through. So I will say that what's wrong with this photo is that we took this just as a demonstration and forgot to put gloves on. You should be wearing gloves when you're doing this, so that's my mistake. But that's essentially once you've filtered your water, use forceps to pull the filter out of the bottom casing. You can see this blue thing here. This is where the cup was sitting on, and then the filter was sitting there. And then you fold it up and put it into the tube. And then in the tube, what we have here is 95% ethanol, which is a good storage medium for, especially if you're in the field and you can't get into a freezer right away. Although if you're going to extract from the filter immediately, you don't have to put it in ethanol first. You can just move it directly into the extraction process, or maybe even put it into lysis buffer, depending on your exact extraction protocol.
Steve?
Once you're in the lab and you've filtered your water or you've centrifuged your water and you're ready to go, again, QA/QC is so critical in working with these samples. It's so easy to contaminate with other DNA if you're not careful. And so I highly recommend that all DNA extraction and PCR setup, all the lab work that you do to set up these samples is done in a clean room. And what I mean by a clean room or a clean lab is an area where there is no PCR product, or you've never brought any animals in that you're working with, or there's no samples that come from really high quality tissue where there might be a lot of DNA there. You're basically keeping out anything that might contaminate your samples of the same DNA source. Ideally, you have something like a laminar flow hood to work out of. That's not critical if everything else is followed, but it really helps out. This is really important. You never ever want to do eDNA in a lab space where you've worked with either the animals themselves or lots of DNA, or have thermocyclers for running PCR in that lab, because it's just way too easy to contaminate. There's a number of different ways you can extract DNA from filters or from the centrifuge samples, but it typically just follows slight modifications of standard methods. So if you're familiar with different DNA extraction techniques like the QIAGEN D&Easy kit, that's what I tend to use, a modified protocol of that. But you can also use other methods as well, like phenol chloroform is a pretty common DNA extraction method that folks use. So it usually use a little bit of modification, but it's not anything too drastically different than any other type of DNA extraction. In my case, I tend to, with the filters, tear them in half. so I have a backup in case something goes wrong with the first extraction. Dry them. If you do store them in ethanol, it's really important that the ethanol dry off before you continue with the extraction process, but then go through the kit protocol with some modifications. And of course, all these details are in some of the published papers that have done this. I mentioned field blanks. You also want lab blanks, so lab negatives. So these are samples that you treat the same and have all the same reagents as your unknown samples, but that you just don't put anything in there, or a blank filter or something like that. So again, it's a way to make sure that you don't have contamination in your lab environment. This is good practice for any sort of genetic work, but it's especially critical for eDNA. So I'm going to transition to once you've extracted the DNA and you're figuring out, okay, well, I presumably have DNA extracted from my source. How do I actually see if it contains DNA from my species or species of or multiple species of interest? And there's three kind of broad ways that folks have detected DNA of species. One is just kind of using traditional PCR presence absence. So if you're not familiar with PCR, it stands for polymerase chain reaction. Basically, in a nutshell, it's a way that we can artificially replicate DNA in the lab. And so we can make lots and lots of DNA from a particular fragment. And we define that fragment, that amount of DNA that we're replicating, by what are called primers. And primers are basically the bookends for that sequence. And it's what directs the PCR process, that you're only getting, or at least ideally, only getting fragments between those two primers. I'll talk a little bit more about primers in a moment. So one is just running a standard PCR, and you can run it on agarose gel and see, do you get those fragments or not? If you do, it means it suggests it's positive. If not, it suggests it's negative. Middle is quantitative PCR, which is a really common now way of looking for a particular species. It's, again, using the PCR process, except you have an additional probe component that I'll talk a little bit more in a moment, where you can actually quantify the amount of DNA that's in there. So you can not only get presence absence, but how much is there. And then finally, metabarcode sequencing. So this is, you're actually sequencing the DNA within your sample, and this is commonly used to identify multiple species within a sample of a community. So I'm going to talk just a little bit about these specifically. So for the PCR presence absence, this requires species-specific primers. So what it means is these are the sequences that bookend your fragment of interest that match up with whatever species you're interested in, and don't match up with species that you're not. So you have to know what you're targeting. You're not going to use this sort of PCR presence absence for trying to get all of the species within your water sample. It's very inexpensive to run. I mean, you could, if all you're doing is running a PCR and then running it on agarose gel, which is pretty common equipment to most labs, I mean, you can do it for a few dollars per sample. It's not very much. You could potentially multiplex it if you use fluorescent primers and then run it on a sequencer using fragment analysis like you would microsatellites, but you wouldn't multiplex if you were just doing an agarose gel. And that adds a little bit to the cost, but it's still relatively cheap. And when I'm throwing out some of these cost generalities, I'm not including things like labor. I'm just talking about supply costs. So this is really definitely a good way if you're just getting into a system and want to investigate how things are working, but it doesn't necessarily have as much information as some of the other methods, as we'll talk about. So qPCR, quantitative PCR, is also a species-specific method. So again, you have to design primers that fit to whatever species you're targeting. It's not going to get the whole community. In what qPCR adds, well, number one, you have different equipment to run it on. The previous one, a standard thermocycler will work. The qPCR has a special machine that's made to, a special cycler that's made to pick up these fluorescent probes. And what the fluorescent probe does is it's a short sequence in the middle of your fragment that you're trying to amplify. And every time your DNA is replicating in the PCR, that probe fluoresces. And so the sensors on the machine will pick that up. And if it's not amplifying, if it's not replicating, it won't fluoresce. So it'll just be like, if you can see at the very beginning, before on this graph, before you have any amount of DNA replication that is that the machine can pick up, you see it's a flat line. And if that flat line continued across the entire graph, that would be a negative. There's no fluorescence that can be picked up. But as you start to get enough amplification, enough replication that the machine can pick it up, you start to see an increase in fluorescence, which is essentially what this line is picking up. And this tells us, and this is a really standard, you have this exponential increase and then it plateaus off. This is telling us that this is a positive sample. And if we have standard curves, so if we have standard samples that we know the amount of DNA is in, we can extrapolate how much DNA is in our unknown samples. We can easily multiplex at least 2 to 3 species, depending on your real-time machine, maybe more, by giving them different fluorescent colors on their probes. And this is a really common way that a lot of folks, including myself, are amplifying eDNA, particularly if we're focusing on species or a small group of species. So I've talked a bit about species-specific primers. Just to emphasize, this is a really, really important aspect if you're trying to identify a particular species, because as you probably can tell, neither of the previous methods are you sequencing anything. So you have to trust, you have to be confident that when you see a positive band or that amplification, that it's only amplifying the species you're interested in. And the way you gain that confidence is by first designing those primers so that the 2 bookends match up perfectly, or at least very nearly perfectly, with DNA from your target species. So this requires that you have sequences from whatever you're interested in to begin with. And there's a lot of, particularly for North America, there's a lot of sequence data already on the web. So this doesn't mean you have to go out and get your own sequence data usually, especially for North America. And it needs to have multiple mismatches of nucleotides with other species that might be out there. So it doesn't bind with those and it doesn't replicate those species. And so you can look on the computer to design primers that based on sequence data that look like that should fit that. Then you test that in silico on the computer to say, okay, it looks like they shouldn't amplify the other species. So if I'm interested in spotted salamanders, if I design it right, it shouldn't amplify Jefferson salamanders or wood frogs or whatever. Of course, then you need to actually test it more than just on the computer. So testing in vitro would mean that you would get DNA samples from these different species and run your protocol with those DNA samples and make sure that it doesn't amplify them. If it does amplify them, then you need to go back to the drawing board to make sure that ultimately the one that you deploy on unknown samples, you have as confident as you can get with these trials that it's only going to amplify your species of interest. Now, you don't have to worry about that with metabarcode sequencing because you're trying to amplify a large number of species within a community. And that community is defined by the primers that you use. So you're still using these primers, these bookends of the fragment you're trying to amplify. But in this case, for multiple species, they're what's called conserved primers. So they're ones that are going to match up, or at least be closely matched enough to bind to a whole host of species. So say you're trying to amplify all amphibians. You can design primers that match up with most amphibians, but then are variable from, say, fish or invertebrates or whatever. So you're going to get more species, but you still have to do that primer design. For this, we're using next generation sequencing platforms where you can, especially with barcodes, you can put on unique barcodes on these different samples, and you can run a lot of different samples and for a lot of different species at once. And so this graph here is just showing the number of sequences, which is also called the number of reads. And the colors aren't important. They represent different species, but you can see multiple colors, which means that, and these are all different sites from this particular study, that they picked up multiple species. Although in this case, whatever the purple species is, you can see it is really dominant. So this is a really good platform to use if you're interested in the community. I should mention people are interested in cost for the qPCR, which I just talked about, the species-specific one. Again, supplies only. Generally, at least like the protocols that I use, $15 to $20 a sample for the reagents to run those, again, not including things like labor or overhead. Metabarcoding is certainly more expensive than that, and really is, but it's becoming much cheaper, and particularly if you're running a lot of samples at once, or you're working with other labs that are running samples at the same time, because on these next-gen platforms, you can run a lot of different multiplex, a lot of samples at once. And so a next-generation sequencing run on its own is several $1,000s, but if you're running a lot of samples or sharing it with other folks, the cost per sample can go down quite a bit. So that's really variable just depending on the situation in which you're running them. So that's, I know very quickly in a nutshell, how we actually get a species ID, and in some cases, the amount of DNA from a sample. So the rest of the time, what I'm calling frequently asked questions. So a lot of these are based on questions that were forwarded to me by Jen that she had heard from other folks, or things that I've gotten just from talking to people. I'm having case studies kind of mixed into the answers to these questions. And what I'm hoping, again, that we'll have a little bit of time that you can ask your specific questions about either the methodology or what I touch on now.
Hey,
so the
Steve.
first obvious question.
Steve,
yeah, go ahead.
Would it be okay if we went through some questions that people had specifically about the content you just covered really quickly?
Yeah, sure, no problem.
Okay, and I apologize if you did answer the question after they asked it. I've been navigating the questions as they're coming in, and so haven't been able to devote my full attention to the presentation. But the first question is, what do you mean by a field blank?
Sure, that's a good question. So a field blank is, as I mentioned, some kind of clean water. So ideally deionized water, but if you can't get deionized water, some water source where you're absolutely sure your target species wouldn't be in. And you would say let's say you're filtering in the field. Maybe you have a large bottle of your deionized water. You would bring with you out in the field. You filter, say, 3 samples from a river. And then right after, either right before or right after, depending on how you're doing it, you would then pour your clean water through that same filtering apparatus and then save that filter just like you were going to try and analyze as an unknown sample. And that way you know if in between your samplings, in between sites with your equipment, if you're getting contamination. Because if your field blank, when you've just poured in the DI water, shows up positive, then you know there's a problem. And there's different ways of doing it. Like, let's say you're bringing things from the field back into the lab. You may want to still fill up, take your bottle out in the field, maybe even roll it around. Like if you're storing all your samples in a cooler, store it in the same cooler. So if there's contamination that's occurring between samples on the way back to the lab, you're picking that up too. But it's anything that, the only difference between how you're collecting in the field is that it's water that doesn't have any target DNA in it.
Okay, thank you. And then the next question is, why does the filter have to be dried?
If you're using ethanol, it has to be dried because ethanol will interfere with the extraction protocol from the get-go. At least the one that I use. So that's the only reason. It's not that it has. If it's like wet from water, obviously that's fine. It just has to be dried if you store it in ethanol.
Okay. And it says, Steve mentioned that you store the filters in 95% ethanol. Can you also store them in a -80 freezer until you're ready to extract?
Absolutely. Yeah. So with that, it was more of like if you're in the field without easy access. But yeah, the best way is if you have access to it, is to get it right into a freezer. And so if you're filtering in the lab, particularly, and so you put the filter in a tube and then you can turn around and put it in the -80. You don't need to put in ethanol or anything else there. You can just put it right in the -80. But obviously, if you're in the field, that's not a possibility. You certainly don't want to just put the filter in the field, put the filter alone in a tube, and then put it in the -80 2 days later or whatever. But yeah, definitely freezing would work. And in fact, when you're out of the field, even when it's in something like ethanol, you don't have to put it in a -80, but I always put mine in a freezer because that, again, is just going to help be a safeguard for things not breaking down.
Okay. And do you need a clean room if you have a flow hood, or do you need a clean room with a flow hood, like both together?
Ideally, both together. A flow hood on its own is certainly better than nothing. I would still be pretty nervous if I had a lab where I was running PCR product, or I had the tanks with the species of interest, and I was just assuming the laminar flow hood would kind of take care of things. I mean, it would make me nervous, I suppose, you know, if you were very, very vigilant about UVing the hood and all sorts of, but I mean, I wouldn't want to do it. You certainly, I mean, if you're ever in a situation like that where you want to try these samples and you think that you think it's not ideal, but you think it might work, like make sure you just include tons of controls. But like I said, I'd feel very uncomfortable working in a lab that had a lot of PCR product of my species of interest, even if there was a hood in that same room.
Okay. And it says, Steve mentioned that the filters could be stored in ethanol when a freezer wasn't available. Would a conventional freezer work, or is a -80 necessary?
Yeah, for the filters that are in ethanol, a conventional freezer is typically fine. Yeah, I mean, I would say that would be fine for that stage.
Okay. It says, do species-specific primers only target mitochondrial DNA, or do nuclear primers exist or work? We've been having trouble with mitochondrial introgression between two of our species.
Yeah, that's a good question. One thing I didn't really talk about, the source of DNA. In theory, yeah, species-specific should be able to work with any type of DNA, and mitochondrial is by far the most common for eDNA, and the major reason for that is that we have a lot more mitochondrial DNA in our cells than we have nuclear, because we just have one nucleus and a lot of mitochondria. And so when we're dealing with, you know, low copy samples, it's just easier to amplify mitochondrial per se. So if you can, so it probably is going to take more optimization to get nuclear markers to work, just because there's less nuclear DNA. But there's no reason in theory it can't, if you can get those nuclear markers to amplify.
Okay, and we have tons of other questions flowing in, but maybe this next section that you have on FAQs, we'll get to them, and then whatever we can't cover, we'll hopefully get to folks later.
Yeah,
Thanks, Steve.
yeah. Okay, sounds good. So the first question is, of course, does it work? And I mean, you probably figure it does if we're doing this webinar, but I just, and this is off the top of my head, so there's very possible a herp family that I left off that I just either forgot about or haven't seen the paper yet. But this is just a list of different families of herps, and in a couple of cases, a couple of diseases that affect amphibians, that at least one member of that family has been used successfully in eDNA studies. So lots of amphibian, both frog and salamander groups, is it chytrid and ranavirus, a few different turtles, particularly the ones that are semi-aquatic. I'll talk a little bit more about soil, and there's been a recent study that was a snake example for clubrids and pythons. So it works across a lot of different species that are broad ecologies. Now, some of these are more easy than others, and you may have different protocols as far as sampling, but it's been shown to work pretty broadly in a lot of different herp species. So the follow-up question is, okay, you can get it to work, but is it reliable and efficient? And so there's kind of three sub-questions within that. Is there high detection probability? So if you go out and sample water, how often are you going to get a positive if they're actually there? And kind of related to that, is it effective compared to other methods? So yeah, maybe we can get it to work, but if we go out and dip net, are we going to have an easier time to get the same answer? And then of course, you know, how much are false positives a big deal? Are we going to get positives that actually aren't true? So I'm going to break down these three with a little bit of data. So detection probability. So detection probability, for instance, if you're familiar with occupancy modeling, is a key parameter in that. And so I'm just going to show a couple of studies that I've been involved with that get at this. So this first one, this is a Hellbender study that was done by a master's student at Appalachian State, Tommy Franklin, that I helped out with. And don't worry too much about all the graphs, but what the top part of this graph shows eDNA surveys, and the bottom part shows traditional surveys, which in this case was rock flipping and snorkeling for Hellbenders. What I want to point out is that at low concentrations of eDNA, we get close to 100% detection. And in fact, the overall detection rate for eDNA was 0.9 in the system, so really high. It actually was also fairly high with the rock flipping surveys, but it's a lot less effort to collect eDNA for hellbenders than in less habitat disturbance. Even bigger difference is this is an example by a study by Todd Pearson with a very small plethodonid salamander, Urspilurpes brucei, the patchnose salamander, where he did both eDNA and leaf litter bags in the streams. This is an aquatic breeding plethodonid, headwater streams. And he found, again, this is showing pretty high detection probability for eDNA and much lower for litter bags. And you basically, for 2 eDNA samples, that level of 95% detection probability, you need to have 62 nights of litter bag surveys. So it's showing that high detection, and also in this case, compared to the common survey method for the species, much more effective. Similarly, with the bullfrog system that I mentioned from the get-go, this is also in France. This figure shows on the left are surveys that were done traditional, I forget exactly, I think like DipNet, maybe calling surveys for bullfrogs, but traditional field surveys. On the right, the black indicates positive, the white indicates negative. On the right is eDNA, and you can see that there are a lot more black circles with eDNA. So again, in this system, eDNA was a lot more effective. This is not to say that in every single eDNA example that this is the case, and there are some examples where it's just, particularly where field methods are relatively easy to implement and pretty effective, or it's just a highly detectable species, maybe one that calls a whole lot. Like I probably would never bother doing eDNA for spring peepers, for instance. So it's not always more effective, but I think there's a lot of studies that show that it can be more effective than field surveys. Of course, you're not getting your hands on the organism, so there's a lot of information that you don't have. So I see it as complementary to field surveys, even when it has a higher detection probability rather than a replacement. And then false positives, obviously, false positives are a little bit hard to definitively demonstrate without real controlled systems or controlled studies. False positives don't seem to be, at least in my work with eDNA and looking at different papers, I think in general, false positives are not prevalent in these studies. There's not a lot of examples of places where you know for a lot of survey work that they're not there and then you get them. But there are a lot of examples of areas with limited survey work where you get a lot more eDNA positives than survey positives. And so that does speak to the fear that, well, what if some of those are false positives? The best way to try and detect that if it actually is happening is, of course, the negative controls. So if you're getting a lot of positives in your field samples, but you're also getting positives in your DI controls, well, yeah, that's probably false positives. Doing a lot of PCR replicates. So I typically, like for quantitative PCR, I'll typically run them in triplicates. So for each sample, I'll run them 3 times. And so if I just get 1 out of those 3 showing positive, I'll typically rerun them again to make sure I get consistently at least 1 out of 3 showing up. And sometimes you can get 1 out of 3 just if it's very low concentration. That's all you can get. But if I get 1 out of 3 and then I run a couple more times and I'm getting 0 out of 3 on those following ones, then I'm less likely to consider that a positive. So PCR replicates can help with that. If you're always getting it popping up and your negative controls are negative, then you have, I think, more confidence that it's not a false positive. And also spatial replicates within a site. So taking multiple, or I guess maybe I should say site replicates, taking multiple samples within the same site. And if you take a bunch and only 1 replicate pops up as positive, then that might be a false positive. So those are ways to try and, you can never like obviously completely eliminate any chance of it, but those are ways to try and to address it or eliminate it. So can I get eDNA from soil? So a lot of questions I have, including myself as someone who also works on snakes, who's wondered about that. And there's been, I showed that study with the game park. There'd been some work showing carp eDNA from sediment of rivers, but with herps, there's a study that came out this year by Kucharenko et al. Of South Florida, and they were looking at, they did a lab test with corn snakes and then looked in the field for gopher tortoise burrows where, based on telemetry, they knew Burmese pythons were, or they knew that they weren't based on telemetry. and seeing if they could pick up Burmese pythons and in the lab corn snakes. And they found that in the lab setting they could detect DNA from corn snakes, and this figure is showing with the Burmese python. So they took samples at the apron of the tortoise burrow and then down inside. And what they found, what this is showing in a nutshell, is based on their telemetry work, and they were just using presence absence of PCR. They didn't find them anywhere on the aprons, but at sites where they knew pythons had gone into, based on telemetry, they actually detected eDNA successfully inside the burrow. And they did not detect any eDNA in burrows that they had no evidence that pythons were using. So this is really cool, and I think the first, at least published, example for things like reptiles that we can potentially use soil to detect their presence. So do I need my own lab? Obviously, having your own lab helps a lot. And everything I've talked about so far is predicated on either having your own lab or working with a lab. But what's interesting is that there is some technology out there where you can run your own eDNA now and you don't need your own lab. And so this is companies Biomem and Smith Root have developed a portable eDNA system that includes both a extraction kit that can be done in the field, takes 5 minutes from a filter sample, and a handheld quantitative PCR unit, so you can run qPCR in the field. And I actually got to do a test of this with the Hellbender. They've developed a Hellbender kit, and so I did a test using water extracted from our Hellbender head starting center here at the Wilds. And so we did different dilutions to see what was the limit of detection, and then also quantifying it to see, is it as good as doing traditional benchtop lab qPCR. So I ran the samples the way I normally do it, and then I used this portable qPCR method. And what I found, so there's a couple of things to unpack on the slide that I'll go through quickly, but I found that it did a pretty good job. So the top here is the benchtop, so the traditional like lab qPCR. The bottom is the portable qPCR that you can do out in the field with the equipment that they have. And I found that could detect Hellbender eDNA with both of them, and the limit of detection was about the same. So it wasn't that one was greatly more sensitive than the other. And they both quantified pretty well. So this is showing the different dilutions and a regression line showing how well the estimated amount of eDNA followed the known dilution. And you can see that regression line is a little bit stronger. The R-square is a little bit stronger for the benchtop, but it's significantly correlated with both. So it does suggest that you can both detect and quantify for, again, this is species specific, but in the field without having your own lab. Now this, as you might expect, because of the speed and ease of use, the extraction kit is more expensive than per sample than doing like the QIAGEN in the lab, assuming you already have the lab infrastructure. I believe, if I'm not mistaken, it's about $20 a sample for the extraction. And with a portable qPCR unit, you can only run 3 samples at a time compared to 96 in the lab. But if lab space is an issue, or you're wanting to be testing out a protocol, this is definitely a good method for that. And you can go to their website for they have a eDNA water sampler, some other cool stuff available. So what about PCR inhibition? And maybe you haven't asked this question because you don't know what PCR inhibition is, but those who have dealt with it certainly know. PCR inhibition is really any interference with the ability of your DNA to replicate that fragment in PCR. So essentially, it's anything that would cause PCR to not work, even if you have the target DNA in there that should match up with your primers. And there's a whole host of ways this can happen, but it's really common in environmental samples that have organic substances that interfere with the PCR amplification of the PCR replication. And well, what are we dealing with? Well, we're dealing with environmental samples. So we often have to deal with this, particularly really samples with a lot of sediment. And so I did, this is a joint project that I worked with Anna McKee at USGS and also Todd Pearson, where we had sampled eDNA for coastal plain amphibians, so flatwood salamanders, gopher frogs, and striped newts, in wetlands that typically are, highly or pretty acidic, have a lot of organic materials in there. Like they're very prone to inhibition. And in fact, we found that while certainly not every sample was inhibited, we had some level of inhibition across the board in most of the different sites, about a third of the wetlands, and a couple of sites, 100% of the qPCRs were inhibited. So in that case, we would get a false negative if we didn't account for this, because we would see nothing happens. We would assume the species isn't there, but in reality, it could be there. We're just not being able to amplify it. And there's ways to put internal controls into your PCR reaction, and you see if those internal controls don't work, which should work every single time, then you know you have inhibition. So what we did, and again, just going to breeze through this, but we did 3 different methods for trying to get rid of inhibition. So we did dilution, which is a pretty common, cheap way of trying to basically dilute out the inhibitors. And then there's a spin column that removes a lot of the inhibitors. And what we found, what this is showing is how consistent the quantification was. So we found actually that all 3 methods worked, but the closer these 2 are to 0 show like how more on the right, the standard deviation lower shows more consistency. And so the column purification had more consistency from sample to sample compared to the dilution treatments, although those seem to work as well. So there are ways to remove inhibition. Now, these may not work all the time, particularly if you have really bad inhibition, but they seem to be mostly effective. And for studies that we knew had evidence of inhibition, we would just run everything through the spin column. Another common question is, okay, so I pick up eDNA. Well, how long does it persist in the environment? Like, how do I know that it's not something that's been there years ago, or or how recently has that been there? And particularly with water, lots of things can degrade DNA in that environment. So UV light, high temperature, low pH, different enzymes can all break down DNA. And so there's been a few studies that have looked particularly in water, but I'll give a soil example too, to see how quickly DNA degrades after the source is gone. So these are mesocosms with spadefoot toads and newts, and where you see this big drop right here, this is when they removed those animals from the mesocosm. In this case, within a few days, all traces of eDNA were gone, so it didn't last very long. This is another study with bullfrogs and sturgeon. A little bit less steep of a drop-off than before, but you can see the axis is 30 days here, so within 20 to 25 days, you're not picking them up. This is a study by Pillier et al. With in a well-researched stream salamander system, Idaho giant salamanders. They actually put a salamander in a creek that they knew didn't have any, and found that 24 hours or before, in 24 hours, you could pick it up pretty well. You remove the salamander, and almost immediately, this is hours down here, you lost all trace of it. And then actually in the soil study I talked about, the Kukurenko et al., they, with the corn snake in the lab, they removed the corn snake and found, at least in that system, within a few days, within 4 days, the black dots here actually represent eDNA positives. The circles represent something else. They couldn't detect the presence of the snake from the soil. So in general, it doesn't seem to last very long, so it's a very contemporary measure. I will say, even though that soil example showed that it disappeared pretty quickly, that snake wasn't on that soil for a super long time, and people get ancient DNA from soil. So particularly if you have a burrowing species that's producing a lot of DNA in that soil, you might have much longer detection times. And the truth is, for things like herps, we have no idea how long that is. And that would definitely require some study if you were starting a soil eDNA study with some type of burrowing reptile or amphibian. Okay, so only a couple more questions to get to, and then can get to your questions. So one of the million-dollar questions is, well, okay, this is all great if we know they're there or not, but how do we know the population status? So can eDNA estimate abundance? This is what a lot of us would really like for monitoring. And the answer is, well, maybe, sort of, depends on the system. So in more controlled settings, so this is a natural pond, but one that had been censused quite extensively. We see a significant relationship between the density of individuals and the amount of DNA. It follows pretty well this curve for spadefoot toads. It's still significant for newts, but as you can see, there's a lot of noise around that line. This is that Pacific stream salamander system that I mentioned before by Pillier et al. So Idaho giant salamanders and Rocky Mountain tail frogs. Again, positive significant correlations. A lot cleaner pattern with the frogs than you get with the salamanders, but still it follows density. And again, this is a very well-studied system where they had confident estimates of density. I've done some work with hellbenders, and this is just one example. I found that I haven't found a really good relationship with hellbender survey density and eDNA, although I fully admit that it's a lot harder to get good density measures for hellbenders because they're a lot harder to survey for. And in fact, there was a recent study with hellbenders in the Northeast that some colleagues did where they did find at broad scales, it looked like there was a relationship between them. So it does look like in general, you can get a significant relationship between eDNA and density, but we're not to the point where we can have a lot of confidence in saying, okay, we got this amount of eDNA, that means that's X number of individuals. And that's definitely something folks are still working on. Oh, and then if you're doing metabarcoding, you might think the number of sequences, so the number of reads you get, might correlate with abundance. And this is actually a study with fish, not with herps. But they found that if you just looked at the number of reads themselves, or the proportion of the eDNA that represented different species, it didn't have a linear relationship. But if you looked at the abundance of number of reads, it did follow with the rank abundance of biomass. So again, it suggests that perhaps the number of reads can help us at least get an approximate of abundance. And then finally, a lot of work I do is in rivers. Rivers, water's moving, eDNA is going to move. So I get a lot of questions like, well, if you get a positive, what does that mean? I mean, is it right there? Is it kilometers upstream? Like, how precise is your eDNA sample? And so there's been some work in different systems. So I'm referencing, this is the Idaho giant salamander system again. Again, well-characterized densities. So this background gray line represents the density of salamanders. The dashed black line represents eDNA amounts. And you can see that there's a little bit of a lag, but the amount of eDNA does follow these peaks. And there's a lag, but it's pretty short. This is in meters, hundreds of meters, excuse me. So it's showing that here it's probably relatively precise. Of course, these are smaller systems. They aren't large rivers. Kind of on the other hand, this isn't a herp study, but it's with macroinvertebrates. It's a system where they have these lake macroinvertebrates and they sampled downstream where those species shouldn't be. So any pickup would be drift. And they found for some of these up to 15 kilometers, for the Daphnia here, they were able to pick up eDNA 15 kilometers downstream. So this is an example where it looks like there is no drift. This is an example with hellbenders that I've done where this is showing we get different amounts of DNA depending on the year. So we get a big pulse of DNA during the September breeding season. Most of the year, there's no correlation of the position in the stream and the amount of eDNA, suggesting that DNA is not traveling very far. But when we had this big pulse during the breeding season in September, granted, these are only 3 points along the stream, so take that grain of salt. But we did see a correlation between how far down the stream and the amount of eDNA, suggesting that when you have lots of it, it probably travels pretty far down. So that's it for me with that before your questions. Obviously, whatever time we have left, I'm happy to answer questions. Here's my email that you can certainly contact me if you need. And yeah, so I'll turn it over to Jen at this point. And thanks everyone for listening.
Yeah, thanks so much to you, Steve. This has been super informative, and you covered tons of material. So I'm sure that everybody's really grateful for all the information you've provided. And so we are at the top of the hour, but I'm going to continue asking questions and continue with the recording, and folks can hop off as they need to. But I'll go through the list of questions that have been submitted, and if you've already adequately covered it and I missed it, then just say it's already been covered. So one of the participants would like to know more about current costs to set up a multi-species invasive species monitoring and detection program for terrestrial or aquatic species. Yeah, I mean, it's difficult to give an exact cost for something like that because there's so many different variables. I mean, it depends on what lab you're working with or who you're working with. As I mentioned, these multi-species require or typically have been done with things like Illumina next generation sequencing platform is a common one. In a single run of those can be several $1,000s, but as I said, depending on
how many samples you're running. If you're working in a lab that's running a lot of these, where you can share lane space, like it can really fluctuate. And if you're, and the other thing to consider that I didn't really highlight strongly when talking about, but beyond just the cost of actually running the samples and the reagents, you also need to consider the cost to develop these. So if you're trying to identify a community of invasive species that there isn't a lot of previous work done on, and you need to maybe design primers and test out primers, you need to do some mock communities. So basically, where you would take DNA of those invasive species, mix them up together in a lab sample, and see, okay, if I run with my primers, am I going to pick up all the species? Test out different concentrations to see how sensitive it is. So I'm not trying to dodge the question. It's just really hard to give like, oh, it should cost this much. So it depends on a lot of factors, and certainly if someone wants to talk more specifically online, I'm happy to discuss more there in the specific situation. But I guess it is important to say that there can be, particularly in systems that you're trying to just start in, the upfront costs can be kind of high, or I mean not like hundreds of thousands of dollars, but certainly thousands, or in some cases, depending on exactly the scope of what you're doing, tens of thousands of dollars in what labs you're working with. And you don't want to skimp on that upfront, because if you don't do the upfront work, right, that optimization, that proof of concept, then you could run a lot of samples and find out down the line that you made a mistake up front. So anyway, that's my answer to that question. Sorry if it's not more satisfactory.
No, I think it's a great answer. You've given them all the different factors they need to consider, and some ballpark figures. So I think that's great. The next question is, do you use multiple filters when large volumes of water are filtered, such as with 10 liters? And if so, how do you process those filters for DNA extraction?
So I tend to not, myself, I don't, but I know folks that have had. There's actually been studies that have used multiple filters, and what some of them have done is actually extracted from each of those filters, and you can sometimes see like certain, definitely depending on what your targets, some targets will be picked up better with different pore sizes. So particularly if you're trying to look at a community thing, it may be worthwhile, sampling at these different pore sizes and then extracting from all of them. I mean, I suppose you could also extract from multiple filters and combine those samples if you're trying to reduce the number of samples you'd run. But yeah, I mean, I guess those are some different options. I typically, for what I've done, most of the sampling I've done has been in rivers with a few different species. My kind of default is, at least what I try out first, is 1 liter, and in most rivers, as long as they're not super silty, that seems to do reasonably well. But it's certainly, I've had to ramp it down like that. Southeastern coastal amphibian study, we were doing 250 mils, and we're happy with being able to do that. So doing multiple filters is a good way to try and maybe get rid of some of that sediment, but pick up some of the smaller cells or DNA.
Okay, thank you. The next question is, and I think that UT means University of Tennessee, but I'm not sure. It said, has anyone tried using digital PCR? We had biorad reps at UT last week who said it would be a good option for dirty, low DNA concentration samples.
Yeah, so digital droplet PCR is something that fell out in my just being general and not going to the specifics. But it's definitely, it's in the class of quantitative PCR, where you're trying to get a good estimate of the absolute amount of DNA that you have, and it's a much more precise way. I haven't used it myself, but folks have, and it's definitely a much more precise way of quantifying DNA because it's running basically, each one's like a different PCR reaction instead of one larger one. I don't know about it with inhibition one way or the other. It's not anything that I know anyone has specifically worked with or seen, but I have, if the rep said that there's evidence to suggest that it does well with inhibited samples, I have nothing to say that it doesn't. I just don't know.
Okay. Then someone said, I am aware that this is mostly focused on freshwater habitats, but do you have any suggestions for the maximum amount of water to filter for marine environments?
I'm off the top of my head, there's a number of papers with marine systems. I don't know if there's been anything herp-wise, like for sea turtles or whatever, but certainly for fish and in other marine organisms, there's been a number of papers. Because I haven't done marine stuff myself, I haven't paid as much to the details of what they've done, so I actually don't know off the top of my head how much water they filtered. But certainly I can pass on those references or look it up later and see. But there's definitely work that's been done in the marine environment that could guide that.
Okay. And I don't know if you answered this one yet or not, but it says, how long can filters be stored in 95% ethanol, and will they start to break down?
I mean, they could, in theory, they could be stored indefinitely. You know, especially if obviously you make sure the tube is tight, things aren't evaporating, or you store it, you know, you put it into a good freezer afterwards. I mean, it shouldn't break down at all. As far as like, if you're talking about breakdown, like physical breakdown of the filter, the filters I use, so some people have talked to me on how the filters were actually dissolved in the in their storage. I've actually, with the ones I use, that doesn't tend to happen. But even if it does, like, it's still all within that tube. So as long as you have the appropriate preservative, and ethanol is not the only thing. There's other ways, like there's a buffer called CTAB buffer that a lot of folks use. So I'm not trying to suggest that that's the only way to do it, but it should last for a long time, certainly within the scope of when you'd probably do a study. So it really is good opportunities if you're out there in the field and you think you might want to look at eDNA for either a species or community down the line, but you don't have the funding yet, but you're out in the field then, and you can at least like either collect water or filter it, then by all means do that, because you can store these samples. And even if you're looking at a target species now and think, you know, 5 years down the road, you know, you've archived your DNA in a freezer that you use to, say, look at bullfrogs, and you realize, hey, there's, you know, this disease that's emerging, and I wonder if it was at my site 5 years ago, you know, you can go back and test that. So yeah, you definitely want to archive these things in good freezers, and you can come back to them later.
Okay. And then the next, there are several similar questions, but I think that they've been answered. Another question is, do you have examples of collection protocols and lab protocols that incorporate your recommended QA/QC measures that you can share with us through email after the webinar? I know you have that paper. Is it okay for me to share that with the group after this webinar?
Yes, yeah, absolutely. And there are good protocols that I didn't put together myself, but colleagues have, particularly through USGS, and that I can pass on those protocols, both for collecting eDNA and QA/QC. Another good, and you have the critical considerations paper, I think, that you can pass on. That's a good review of some of the things, thinking about designing a study and quality assurance and that sort of thing. So yeah, there are definitely good resources. Fish and Wildlife Service is another one, particularly for their carp eDNA sampling. They have a protocol there, so I can pass on some of that.
Okay, great. Do you have thoughts on ethanol versus Longmire's buffer for filter storage?
I've never used Longmire's buffer myself. Ethanol's worked well for me, but I mean, really anything that is going to that can preserve DNA and is stable over, you know, that's stable, I guess, it should be fine. So there's a whole host of potential preservatives that could be used. I'm certainly not endorsing one or the other method, but I've used ethanol. It's worked well for me, so I've continued to use ethanol.
Okay. One of the webinar participants said that Longmire buffer works well for long-term storage at room temperature. So thanks for sharing that. The next question is, I've heard that people use silica beads to dry out their samples to preserve until they can get it back to the lab. Is that not adequate enough? Is ethanol required?
Yeah, people definitely use silica beads for a number of things. I mean, could be wrong. I don't remember anyone. I don't know of anyone directly that's used for filters, but off the top of my head, I can't think of a good reason why that wouldn't work. We use it for all sorts of other like other tissue samples and things like that. So I don't want to give a definitive yes or no on that, because someone may point out like an obvious reason why that could be problematic. But at least conceptually, to me, that seems like that should be fine. And it's quite possible folks have done it. So I don't know if anyone is on the call still that's worked with it and can chime in, and I can look into that. But it seems like that it could work okay.
Okay. And then we had a request to list the primary labs that you would recommend for an organization that's not equipped to do the analysis and that would only be involved in the sample collections and interested in the results. I'm going to compile that a list of labs, Sean, and share that with the group so I can get that to you. I won't be able to do it within the next couple of weeks, but it will definitely be coming down the pike, and I will share it with everybody on this who registered for the webinar. Let's see. And then here's a question. It says, what does a well refer to in eDNA analysis for, it says you referenced it in number of positive brook trout wells.
Okay. Yeah, a well is just what we call, like on your plate, you have the space where you put the samples. We call them a well. So a well basically would, in a context of say like a PCR, would be like one sample, or one replicate of one sample. So if we were running something in triplicates or running 3 replicates for a single sample, that would take up 3 wells.
Okay. Another question is, how do you calculate the LOD?
So LOD is limited detection. So basically, you quantify based on a standard curve the amount of DNA, and you see what's the lowest amount of DNA that you can reliably amplify. So the limited detection is if the concentration is below that, you might not be able to amplify it with your protocol. And there's also a kind of a complementary called limit of quantification. And so there's cases with qPCR where you can pick up presence absence, but you can't consistently quantify it because it's just too variable, because it's too low concentration. And so that could be a separate limit where you can reliably quantify it. But limited detection is just how low of a concentration of DNA can you reliably detect.
Okay. And can you use eDNA surveys for hybrid species?
Yeah, if you can, as long as you can developing primers that are specific to that hybrid species. Yeah, you definitely can. It just, it's just the optimization and making sure that your protocol will amplify that target. But yeah, there's no reason why you couldn't. And I think, if I recall, there actually is a paper out that did look at hybridization, detecting hybridization with eDNA. Although right now I'm blanking on the details of it, but I could look it up and find it.
Okay. And then even if you answered this question, if you wouldn't mind answering it again, let's see. With heavily sedimented water, do you need to collect more or less water?
That's a really good question. And I don't have like a definitive, confident answer to that. And I think it might depend. I mean, one thing that I've wondered is if having more sediment in the water, you might actually have a higher detection per unit filtered, because you might have cells or things that bind more in that sediment. Or, if the sediment's flushed up from the bottom and you have DNA that settled down there and been kind of preserved in there, if you have a higher chance of detecting it with more sediment. But at the same hand, obviously more sediment, it's hard to filter more water, and so it may decrease things. So I don't have a yes or no definitive always this way. But yeah, I think it could go either way. But it's very possible that having a lot of sediment could increase your ability to detect the DNA if it's there, because it's potentially binding to it. I don't know if there's really been I don't think there's been a lot of work looking at that directly. I mean, the closest I can think of off the top of my head is I mentioned that CARC study where they picked up DNA in the sediment of the rivers. They were also taking water samples from that same area and comparing them, and they found more DNA down in the sediment of the carp, even though obviously carp are most of the time in the water column, which suggests that DNA was settling down and being preserved at least to some degree down the sediment. So if you have a system where a lot of that sediment is coming or a lot of that turbidity is coming from the bottom of the sediment being kicked up, then that might increase it. Now, if you have a situation where you just sampled after a high rain event and most of that sediment is coming off the land, then it may be the opposite. So I try as a rule to, particularly if I'm dealing with systems that aren't typically high in sediment, I tend to, for instance, not sample when there's been high rains or the water's high, because that's going to potentially dilute things, or it's really mucky. Obviously, if you have a system that's always high in sediment, there's not much you can do about that. But I try and avoid it if I can.
Okay. Is there an ideal depth from which to collect the samples?
It depends on your study system, but I can tell you sampling for hellbenders, which are species that lives on the bottom of rivers, that I've collected water. I tend to get it for that species, get it from where it's flowing, not from like a stagnant pool. But just off the side of the creek side where the water is relatively shallow, and I can pick them up. So, but it really depends on what you're looking at, like how the water is flowing, for what's ideal. There's no one size fits all for something like that.
Okay. Do you know of any work done with skinks, specifically the Florida sand skink?
I am not aware. That certainly seems like one that would be a natural to at least test for soil, and there certainly could be folks working on it, but I'm not aware of any published study for eDNA with sand skinks or directly of anyone working on it right now.
Okay. Are there general sampling protocols on where to sample? And so more specifically, are there biases associated with sampling the shoreline versus the surface water versus the center of a pond, lake, or stream?
It depends. I've done, for instance, with river sampling for hellbenders, which is what I've done the majority of my eDNA work with. There's been a few times where we've sampled it like across a river, different points within the same area. And in general, for that system, it doesn't seem to matter a whole lot. Like the water's flowing. I think the DNA or cells are all mixing for the most part. Now, if you're sampling, say, like a lentic environment, and you're sampling a breeding pond, say for some ambystoma or whatever, and there's a part of the site where there's emergent veg, and that's where they're breeding, and that's where the tadpoles are, and you sample on the other side of the pond, and it's big enough, then yeah, that could make a huge difference. So you definitely, as much as possible, you want to target where you think within that site you're likely to have those species. And in some systems, you may be able to be more lenient or liberal with that, and in some systems, you may have to be really close. Like I said, there's those pond systems, particularly for if you're dealing with low density species, like say something like flatwood salamanders. If you sample all throughout the pond, you'll probably notice that you're not getting ubiquitous detection. So in that sense, it really does matter, but it may not in every single case.
Okay. And what is the best way to achieve adequate spatial replicates?
I would say, I guess, a pilot study. It really, I think it depends a lot on your detection. So detection is not, I gave some examples where it was pretty high, but I've also done some mud puppy eDNA, and compared to hellbenders, I actually run them in multiplex. Detection appears to be a lot lower for mud puppies than hellbenders. So for instance, when we're doing kind of a broad survey, just trying to pick up hellbender detections, and we're not focusing on any one site, a lot of times we'll just take one per site, because I found that that's typically all I need to detect them if they're there. But if I'm really trying to focus on identifying whether a site is mud puppy positive, I'm probably going to want to take a lot more than that. So it's really system dependent. And the only way you know that is either by previous work or doing your own pilot study. So I would, when you're starting out a new system, I would encourage taking multiple replicates at a site. I mean, a common is taking 3 replicates, but sometimes you may want more than that, depending on, how confident you are in being able to detect the species. And then you kind of figure from there, okay, can I cut down the number of samples based on my detection rate?
Okay, sounds good. So the next question is, you talked about the parameters that would affect how long the DNA stays in the water. Could you expand a little bit more about them? For example, pH, temperatures, et cetera, and how those affect the longevity of the DNA in the water?
Yeah, so in general, and there's been some work, some studies that have looked directly at this, but in general, the higher the temperature, the more quickly DNA is going to degrade. So colder water, it should last longer. pH, more acidic pH will degrade DNA faster. So that's a concern as well. Now, there hasn't, with these sorts of different environments, it's not to say that, say, a shallow, higher temperature, low pH site doesn't mean that you can't get DNA from it. In fact, with the coastal plain work we've done, those sites are pretty acidic and usually, or a lot of times, not particularly deep and higher temperature, and we've been able to pick it up. But it's just going to degrade a lot quicker. And I mean, I couldn't tell you how quickly because we haven't done that work in that system. And with the studies that have, I forget off the top of my head how quickly it changed. So you can detect them, but it is going to degrade quicker, and so it may reduce your detection probability with those things. And higher UV degrades DNA. So if you're sampling on a global level and or maybe even elevational, imagine higher elevation with more UV, you might have a lower amount of or a quicker time for DNA to degrade. So those are some of the common things, I guess, that we can more easily measure and maybe control for that have an effect on DNA. But just because you have those factors doesn't mean that you can't get eDNA. It just means it's probably going to degrade faster.
Okay. Do you have any recommendations on how to estimate statistical power for designing sampling protocols, specifically for mapping amphibian distribution in a watershed?
I mean, so the question is how to for doing a power analysis or something like that?
Yeah, how would you estimate statistical power? Any recommendations for that, for designing sampling?
Well, I mean, I think probably a relatively straightforward way would, as I mentioned, doing a pilot study, particularly in an area where you knew had your species of interest. And then, I mean, the easiest, I guess, easy in the sense of statistically, I mean, may not be easy with the field stuff, but sampling a few well-known areas, as well as some areas where maybe you're pretty sure they're not, and using an occupancy modeling framework to estimate detection probability, and use that to guide what your power is and how detectable it is. I mean, that would be I think the best way to do that.
Okay. And so we have a bit of information provided by one of the participants. It says drying filter samples with silica beads can preserve eDNA as well as ethanol. However, as always, care must be taken to avoid contamination. So thanks, Catherine, for that helpful tip. We have a request from Bonnie for you, Steve. It says resources on species that have existing species-specific primers would also be useful. So if we can share that. We have a request for you to point out the difficulties, it says with bioinformatics, and it says that collecting eDNA is easy in comparison.
Yeah, so that's a good point. Like I said, there's a lot I glossed over just for trying to fit in a lot into this. But what they're referring to the bioinformatics is for the metabarcoding, so for the multi-species. I probably made it seem a lot easier than it actually is, but particularly if you're working in a new system, you have to have a reference database of all the species that you're interested in because you need to know if those sequences match up to it. But there's also a lot of computationally intensive work to even identify those sequences. So it's not something that that without, if you've never done it before, not working with someone who has, there's definitely a big learning curve, and that would be a whole nother webinar in itself. But thank you for pointing that out. I probably made it seem a lot more straightforward, easier than it is. That being said, anyone could do it if they're willing to learn, or they're working with someone that does. But there are a lot of factors that I didn't talk about.
Okay. And another helpful tip from Catherine is that the eDNA group at Washington State University has put together a website that hosts a variety of eDNA resources, including protocols, and she provided that website. And so I will share that with the group as well, Catherine. So thanks for sending that, and I'll send it out whenever the recording comes available. But Catherine says that any contributions of additional resources are always appreciated. So Steve, if you have anything you want to add to that site, it sounds like they'd be, they'd be very receptive to that. Another question is, can you combine your spatial replicates together if you are sampling one water body source, so you only use one filter?
Sure. Yeah, you could, if you're not interested in where they are in there, and you're just trying to get a detection site, absolutely you can. I mean, if you're doing something where you're combining equal amount of water from different places, and let's say you're taking, just hypothetically, from 10 different areas and only one of them have your species of interest and there's not tons of them, then you may worry about, if you're combining water, diluting your DNA that you sampled. But there's no problem if all you care about is detection of the site, combining. And I've done that to some degree for some. You just want to make sure you don't reduce your chance of detecting true positives.
Okay. And then the last question I can answer, it says, can we get the PowerPoint for this webinar at a later date? Well, actually, I was going to say that the recording will be available at a later date, but sounds like she's asking specifically for the slides. And I don't think you had notes associated with your slides. Is that right, Steve? But I guess that's up to you, Steve, if you wanted to make your PowerPoint available to others.
Probably. So it might have noticed on the bottom, a couple of slides I used that were put together by collaborators. I just want to check with them to make sure that they were fine with it before sending it out. But probably. But let me check first.
Okay. And yeah, so the recording will be available. And as well as the other information I said that I'd provide, again, a list of laboratories where people can send samples. That will come in several weeks, so it won't be sent out with the initial recording. And then I'll also include that site that Catherine recommended as well. So thanks so much, Steve. This has been a fantastic webinar. We have, well over 200 participants, lots of great questions. So there's definitely a lot of interest in this topic, and people were definitely pleased with it. And Catherine sent a little note, thanks for a really informative webinar. It's a great summary of eDNA methods and considerations, and some other thank yous are coming in as well. So thanks again, Steve. We really appreciate your time and expertise.
Yeah, well, thanks everyone for tuning in. This was great to do.
Sure thing. Bye.